Right now, the biggest titans in tech, I mean, we're talking massive, well-funded university labs, the billionaires building things like Neuralink, they are spending billions of dollars trying to figure out how to safely jam metal, silicon, and flexible plastics into the human brain.
Yeah, massive amount of money.
Right. They're basically trying to build the ultimate brain-computer interface.
Yeah.
But what if they are all trying to solve the exact wrong problem? Welcome to the Deep Dive. Today we're looking at a paper by an independent Nebraska-based researcher named Zachary David Enfield, who says the future of the mind isn't a microchip.
Not at all.
It's actually a stealthy, quantum-conducting organic protein.
Yeah, it's a complete paradigm shift. Enfield's paper is called The Organic Quantum Interface, and the mission of our discussion today, our whole deep dive, is to fundamentally rethink the entire architecture of how we connect humans to machines.
Everything we thought we knew.
Exactly. We are talking about abandoning the idea of putting wires into the brain and instead looking at how we might grow organic extensions out of it.
Which sounds crazy, right? And if you're listening to this, wondering why the microscopic engineering of a brain-computer interface matters to you, think about what's at stake here.
Oh, everything is at stake.
Right. Whether you are tracking the future of cognitive enhancement or, looking for hope in the treatment of severe mental health conditions, or if you're just endlessly fascinated by the sheer, staggering biological engineering of your own mind, this matters.
It really does.
We are going to break down a reality that sounds like pure science fiction, but is actually grounded in intensely rigorous, peer-reviewed molecular biology.
So I want you to completely erase the image of a metallic implant from your mind.
Okay, so no glowing blue chips.
Right. No silicone grids, no gold contacts, no rigid needles. Instead, imagine a microscopic protein-based scaffold.
A protein scaffold.
Exactly. Something that speaks the exact same chemical and quantum language as your own neurons.
But to understand why Enfield is proposing this wild new organic quantum interface, we really have to start by looking at the current technology.
We do.
Because to read this paper is to realize that the current BCI industry, despite all the flashy press conferences, is essentially hitting a massive biological brick wall.
They really are hitting a wall, and it comes down to a crisis of fundamental assumptions.
How so?
Well, every single brain-computer interface built to date, from the classic rigid silicon of the Utah array to the newer, highly flexible polymer threads we see in the most advanced labs today, they all rely on one core, unquestioned idea.
Which is what?
They all assume that if we just engineer a synthetic material well enough, we can make it biologically acceptable to a living neural environment.
Right. If we just make the plastic better, the brain will like it.
Exactly. And Enfield's argument is just a flat, definitive, no, you can't.
Because the brain evolved over millions of years to do one thing perfectly when it comes to its borders.
Right.
Right? Identify foreign material, attack it, and isolate it.
Exactly. He focuses heavily on the glial scarring response.
Right, glial scarring.
Yeah, so usually when engineers put a probe into the brain and the signal degrades over a few months, they view it as an engineering failure. Like, oh, the probe failed.
The hardware broke.
Right. But Enfield flips that perspective completely. He says, no, the glial scarring isn't an engineering failure. It is a massive, spectacular success of the brain's immune system.
Wow. Let's actually unpack what that scarring process is, because it's not like a scrape on your knee, is it?
No, not at all.
When a wire goes into the cortex, you have these specific immune cells, microglia and astrocytes, that basically act like the brain's bouncers.
That's a great way to look at it. The bouncers.
Right.
Microglia are these fascinating cells. In their resting state, they have these long branching arms constantly feeling around the local environment.
Like little tentacles?
Yeah, exactly like that. And the moment they detect a surface chemistry that isn't biologically native, like silicon or a metal alloy, they pull those arms in, they change shape, and they physically swarm the object.
They just attack it.
Right. They secrete inflammatory chemicals, and then the astrocytes come in and build a dense, fibrous wall around the probe.
It's like encapsulating a splinter.
Precisely.
The brain is literally building a quarantine zone around the implant, which is why longitudinal neural recording, actually keeping a BCI working for months or years to help a paralyzed patient, for instance, is such an intractable problem.
Right, because the tissue is succeeding in its immune objective.
The neural inflammation produces oxidative stress, which then literally corrodes the electrodes. It's like a self-amplifying cascade of destruction.
It's a brutal environment for electronics.
So how has the industry been trying to fix this?
with what Enfield essentially calls expensive band-aids.
Band-aids.
Yeah, it's a pure materials science approach. They try to make the foreign object more polite, essentially.
Okay, polite how?
For instance, they use hydrogel coatings. Think of a hydrogel like a microscopic water balloon made of polymers, like PEG polyethylene glycol.
Okay.
They coat the stiff needle in this soft, wet gel to try and trick the surrounding tissue.
Right, because there is an immense mechanical mismatch between a metal wire and the brain. I mean, the brain is incredibly soft. It has the consistency of warm jello.
Precisely. If we look at the physics of it, a rigid silicon probe has a stiffness what engineers call a Young's modulus of about 200 gigapascals.
200 gigapascals.
And brain tissue is down around 15 kilopascals.
Ooh, that is a staggering difference.
It is. So every time your heart beats, your brain pulses and shifts slightly inside your skull.
Right.
If you have a stiff wire sitting in soft jello, that pulsing creates a micro-slicing effect. It constantly irritates the tissue.
Yikes. So they try to soften the materials?
Yes, exactly. They use flexible parylene C substrates to let the implant bend.
Right.
They coat the electrodes in conducting polymers like POOT.PSS, which is a synthetic polymer that conducts electricity but has a softer, more organic feel than bare metal.
Right. It lowers the electrical impedance.
Exactly. And to be clear, this is all genuine, brilliant engineering progress.
Sure.
But Enfield argues they are fundamentally solving the wrong problem.
Because no matter how soft the plastic is or how much hydrogel you smear on it, it's still foreign.
It's still an invader.
It sounds like current BCI engineers are trying to learn French by just shouting English at the brain in a slightly softer voice.
Exactly.
Or maybe putting on a nice fuzzy sweater while they shout English. The brain still knows it's not speaking its native chemical language.
That's spot on. The brain's native chemical language is aromatic, it's hydrophilic, meaning it loves water, and it is intricately protein-coated.
Okay.
Synthetic polymers and silicon are none of those things. The brain wants a native conversation partner.
Right.
It doesn't want a better electrode. It wants a biological scaffold assembled from the exact same molecular components that it already uses.
Which means to figure out what the brain actually wants, we have to look at how the brain actually communicates.
We do.
And this requires us to zoom way, way in, past the cells, past the synapses, down into the quantum realm.
Yeah, this is where Enfield's paper really takes a radical turn.
Buckle up, right?
Yeah. So the classical model of the brain, the one we all learn in high school biology, involves electrical action potentials, shooting down a nerve fiber, releasing chemical neurotransmitters across a gap, and triggering the next cell.
Right. Electricity and chemicals.
Exactly. But Enfield points out that this is just the macro scale view. It's accurate, but only at the scale of entire networks.
It's the zoomed out map, not the street view.
Right. But if you look inside the neuron itself, down at the scale of individual proteins, the neuron doesn't function like a traditional electrical wire at all.
Okay.
Enfield argues it operates as a quantum optical device.
Okay, I have to stop you there. A quantum optical device inside our cells. That sounds completely wild. Where is this happening?
It's happening in a structure called the microtubule.
The microtubule. Okay.
Right. Inside every single cell in your body, but especially in neurons, you have this dense cytoskeleton. The main components of that skeleton are microtubules.
What do they look like?
They are tiny cylindrical tubes, about 25 nanometers wide, made of a protein called tubulin.
Okay, so like tiny pipes.
Yeah. Historically, biologists thought they were just the physical scaffolding that gave the cell its shape, or, little railroad tracks for motor proteins to carry nutrients around.
But they aren't just structural support beams.
Not at all. They are the densest concentration of aromatic amino acids in the cell.
Okay, aromatic.
Yes. And this is where the specific molecular geometry becomes the most important thing in the world. Every single tubulin dimer, the basic protein block that stacks up to build these tubes, contains exactly 86 aromatic rings.
Okay, wait. Let's clarify a term here. When you say aromatic, my mind immediately goes to like a scented candle or a strong cup of coffee.
Right, yeah.
But in organic chemistry, that means something completely different, right?
Excellent point. Yes, in chemistry, an aromatic molecule has nothing to do with smell.
Right.
It refers to the physical shape of the molecule. It means the atoms are arranged in a flat, stable ring, usually a hexagon of carbon atoms.
Okay, flat ring.
And because of the way the bonds work in that ring, the electrons aren't locked to specific atoms.
Oh, really?
Yeah, instead they form a sort of free-floating cloud of negative charge above and below the flat ring.
So the microtubule is packed with 86 of these specific ring-shaped structures with clouds of electrons hovering around them.
Exactly. Specifically, it's 8 tryptophan residues, 36 tyrosine residues, and 42 phenylalanine residues.
There's a lot of rings.
It is. But Enfield zeroes in on those 8 tryptophan rings. They are the absolute stars of the show.
Why the tryptophan?
Because within the microtubule, these tryptophan rings are geometrically spaced out with center-to-center distances of exactly 1 to 2 nanometers.
Okay, and why does that specific 1 to 2 nanometer spacing matter so much?
Because that spacing mirrors one of the most famous structures in quantum biology.
Yeah.
The Fenner-Matthews-Olsen complex, or the FMO complex.
The FMO complex. Wait, that's involved in photosynthesis, right?
Yes. Back in 2006, scientists made a monumental discovery.
Oh, I remember hearing about this.
Yeah, they were trying to figure out how certain bacteria capture sunlight and transfer that energy to their reaction centers with almost 100% efficiency. It basically defied classical physics.
Because it was too perfect.
Exactly. What they proved experimentally was that the FMO complex uses quantum superposition.
Superposition, meaning being in multiple states at once.
Right. The energy doesn't just travel down a single path. It explores all possible paths simultaneously as a quantum wave, finding the most efficient route instantly.
So plants and bacteria are literally using quantum physics to process sunlight.
Yes. And the structural geometry that allows them to maintain that quantum state, that exact spacing of 1 to 2 nanometers between aromatic rings, is mirrored perfectly in the tryptophan networks of the microtubules in your brain.
Wow. Okay, I'm going to push back here, though, because whenever the phrase quantum biology comes up, the immediate counterargument is always about the environment.
The heat problem.
Right. I've always heard that to build a synthetic quantum computer, you have to cool the processor down to almost absolute zero and isolate it in a vacuum because quantum states are incredibly fragile.
Very fragile, yeah.
Any outside interference causes them to collapse.
Yeah.
So the human brain is notoriously hot, wet, and messy. How on earth does a delicate quantum state survive in a warm, squishy neuron?
That is the exact friction point that kept quantum biology on the fringes for decades. It seems totally counterintuitive.
It really does.
But what's fascinating here is the proven mechanism of phonon-assisted quantum transport.
Phonon-assisted, meaning sounds or vibration.
Exactly. Heat is just molecular vibration.
Right.
In a synthetic quantum computer, heat destroys coherence. But in the photosynthetic FMO complex and in neural microtubules, the biological structure evolved to use that heat.
Oh, wow.
Yeah, the thermal vibrations, the messiness of the biological environment actually sustain and drive the quantum coherence rather than destroy it.
So the noise is a feature, not a bug.
Exactly.
The protein jiggling around actually keeps the quantum wave going.
It couples the vibrational environment to the electronic states perfectly. So these tryptophan rings inside your neurons aren't just sitting there. They are undergoing quantum dipole oscillations at the terahertz scale.
Terahertz? That's trillions of cycles per second.
Trillions.
That is unimaginably fast.
It is. And according to this model, these quantum oscillations propagate through the microtubule lattice in a fractal-like cascade.
Like a waterfall of frequencies.
Yes. They start at the terahertz level, then step down to gigahertz, then megahertz, kilohertz, and finally down into the low hertz range.
Wait, the hertz range like tens or hundreds of cycles per second. That's where we see classical electrical action potentials.
Precisely. What Enfield is demonstrating is that the electrical spike that a current BCi reads, the signal that Neuralink or a Utah array picks up, is just the very bottom of the cascade.
The very bottom.
Yeah, it is the downstream output.
So the classical electrical signal is basically just the printout.
That's a perfect analogy.
Just the receipt.
Yeah. Current BCIs are reading the receipt from the cash register after an incredibly complex quantum computation has already taken place. They are capturing the final end state of a cognitive process.
That's incredible. Imagine trying to understand the plot, the emotion, and the cinematography of a 2-hour movie by only looking at the tiny paper receipt from the ticket booth.
You'd miss the whole movie.
Exactly. That's what we are doing when we stick metal wires into the brain. We read the printout. What Enfield is saying is, let's stop looking at the receipt. Let's tap into the projector. Let's access the quantum optical layer of the computation itself.
But to do that, you can't use silicon.
Right.
You have to physically build a bridge that speaks that exact same quantum language. You have to build an aromatic highway that can link directly to the brain's own tryptophan network.
Which brings us to the actual construction of this organic quantum wire. How do we build it?
Well, Enfield proposes building a scaffold out of an amyloid protein,
Okay.
specifically a protein derived from a yeast prion called SUP35.
Now, when I hear the words amyloid and prion, my mind immediately goes to neurodegenerative diseases.
Oh, sure. Alzheimer's, Parkinson's?
Right. Alzheimer's involved amyloid plaques. Mad cow disease is a prion disease. Why are we introducing these into the brain?
It's a very natural reaction to have, but SUP35 is entirely different.
Oh.
It is a translation termination factor found naturally in yeast. It has absolutely nothing to do with mammalian disease pathways.
So it won't cause mad cow disease?
No, definitely not.
So why use a yeast protein for a human brain implant? Why not use a human protein?
Stealth. Complete immunological stealth.
Yeah.
Because SUP35 is a yeast protein, the human brain has no evolved immune program to specifically recognize and attack it.
Okay, so the bouncers don't know who this guy is.
Exactly. At the same time, because it is made of biological amino acids, it doesn't trigger the aggressive foreign body rejection that rigid silicon does.
A VIP pass.
Yeah, it is biologically recognizable as a natural protein, so it doesn't cause alarm, but it's fundamentally invisible to the specific attack vectors of microglia. It flies completely under the immune radar.
That completely solves the rejection problem we talked about at the beginning. And structurally, I understand amyloid fibrils are incredibly tough.
Oh, they are. Once they fold into their final shape, they have a mechanical stiffness comparable to nylon or spider silk.
Sublayer silk, that's strong.
Yeah, they are durable enough to serve as a physical wire, but soft enough to bend with the brain's natural pulsations without causing that microslicing tissue damage.
No more slicing the jello.
Right. But the magic isn't just the backbone of the protein, it's what Enfield proposes engineering into that backbone.
Right. He doesn't just want to use the raw yeast protein. He wants to modify a specific tiny fragment of it, the CGNNQQNY peptide, by perfectly inserting tryptophan and tyrosine amino acids into its structure at exact intervals.
And here is where the molecular architecture becomes absolute genius. When this specific yeast peptide folds into what's called a beta sheet structure, it naturally spaces its amino acids exactly 4.8 angstroms apart along the spine of the fibril.
4.8 angstroms. Why is that specific number so critical?
Because 4.8 angstroms is the absolute ideal distance for a phenomenon called pi-p stacking.
Okay, let's break down pi-p stacking.
Sure.
Earlier, we talked about aromatic rings having those free-floating clouds of electrons, the pi electrons, hovering above and below the flat ring.
Yes.
When I looked into this, I visualized it like a stack of coins or maybe a stack of pancakes. Is it that the rings sit flat on top of each other? And at exactly 4.8 angstroms apart, those electron clouds kind of merge into one continuous elevator shaft.
That is a phenomenal way to visualize it. Yes, when you stack these flat aromatic molecules like pancakes at that precise distance, those separate pi electron clouds overlap and merge. They delocalize.
And that overlap allows the quantum excitation, the energy, the information to flow seamlessly from one pancake to the next without losing coherence.
Exactly. It creates a low-resistance quantum channel. It's the exact same structural basis used for charge transport in advanced organic semiconductors.
Wow.
Enfield's design inserts a tryptophan molecule at every fourth position in the yeast scaffold.
Okay.
Because of the way the beta sheet twists, this creates a continuous, perfectly spaced vertical column of tryptophan running the entire length of the biological wire.
And tryptophan is special because, unlike some other amino acids, it has a double ring structure, an indole ring.
Right. That double ring gives it a much larger surface area for that pi electron resonance. It is perfectly optimized for parallel face-to-face stacking.
Like the best possible pancakes.
Exactly. Think of the tryptophan column as the primary high-speed vertical elevator for the quantum signal.
But the design also includes tyrosine.
Yes, it does.
And tyrosine only has a single ring, a phenol structure. What's the point of including a single ring if the double ring is so much faster?
Tyrosine introduces a completely different geometric capability. Because it has a hydroxyl group attached to it, it doesn't like to stack perfectly flat face-to-face.
Yeah.
It prefers to interact at an angle in a T-shaped stacking interaction.
So if tryptophan is the high-speed vertical elevator moving the signal straight up and down the wire, tyrosine forms the lateral hallways on every floor.
Yes.
It allows the signal to move sideways between adjacent strands of the protein scaffold.
Exactly. It prevents the system from being just a fragile, one-dimensional wire. By weaving tryptophan and tyrosine together, you create a complete, robust, three-dimensional transit system for quantum information.
That's so smart.
It is. If one pathway is blocked or vibrating slightly out of phase, the signal just routes laterally through a tyrosine hallway.
Incredible.
And we know this works.
Mhm.
Surface-enhanced Raman scattering studies have provided direct, real-time spectroscopic evidence of this exact kind of pi-stacking delocalization in engineered amyloid structures.
It's just astonishing. But there's one more piece to this biochemical puzzle that really threw me.
What's that?
We are talking about quantum electron clouds and terahertz oscillations. But our brains run on chemistry. Dopamine, serotonin, acetylcholine.
The what chemistry?
Yeah. How does this pure quantum realm actually connect to the wet chemical neurotransmitters floating around in our heads?
Through an incredibly profound mechanism known as the cation-pi interaction.
Cation-pi. A cation is a positively charged ion, right?
Yes. And remember those pi electron clouds hovering over the aromatic rings?
The pancakes.
Right, the pancakes.
Oh.
They are negatively charged.
Okay.
So you have a powerful electrostatic binding force between a positively charged molecule and that negative quantum cloud.
Opposites attract.
Exactly.
Yeah.
It turns out that major neurotransmitters like acetylcholine, which is a quaternary ammonium ion, meaning it has a strong positive charge, bind to receptors in the brain, specifically through cation-pi interactions.
So the chemical neurotransmitter literally, physically locks into the quantum electron cloud of a tryptophan ring.
It does. The cation-pi interaction is the primary binding mechanism.
That is wild.
What this implies is that chemistry and quantum physics in the brain are deeply, inextricably intertwined right at the receptor level. The physical binding of the chemical is the transduction event. It's the moment an extracellular chemical signal is converted into an intracellular quantum state.
Okay, so let's take a breath and look at what we have so far.
Sure.
We have the blueprint. We know how to build a stealthy, organic, 3D quantum wire out of yeast prions, laced with perfectly spaced aromatic amino acids that can catch and carry quantum signals from neurotransmitters.
They do.
That's the material.
Yeah.
But the human brain is vast. It has nearly 100 billion neurons.
A very big place.
Where exactly do we plug this wire in to get the most valuable data?
If you are going to build the ultimate interface, you don't go to the motor cortex.
Really? But that's where everyone goes right now.
Most current BCIs go to the motor cortex to read the desire to move a limb, yeah. But that's a downstream command.
Okay.
To truly interface with the mind, you go to the holy grail of human cognition, the prefrontal cortex.
CFC. Right behind your forehead.
Yes. This is the anatomical seat of executive function. It's where working memory lives, decision-making, long-term planning, impulse regulation.
It's essentially where your personality is processed.
It's where a decision is actually made. The motor cortex is just the shipping department where the resulting command is dispatched to the muscles.
I love that analogy. The shipping department.
Yeah. As Enfield notes, an interface reading the motor cortex is reading a letter that has already been written and posted. An interface reading the prefrontal cortex is reading the author's underlying thought as the decision first crystallizes.
But historically, getting clean data out of the prefrontal cortex has been incredibly difficult.
Extremely difficult.
Current non-invasive tech just doesn't cut it. You have things like FNIRS, functional near-infrared spectroscopy, which shines light through the skull to track blood flow.
Right.
But tracking blood flow is like tracking a metabolic shadow. It lags 2 to 6 seconds behind the actual thought.
And 6 seconds is an eternity in cognitive processing.
Right. And EEG, measuring electrical fields through the scalp, is just horribly blurred by the thick bone of the skull.
It's like trying to listen to a conversation from outside a concrete building. To get high-fidelity data, you absolutely have to get close to the neurons.
But penetrating the prefrontal cortex with rigid electrodes is highly dangerous.
Very. It carries enormous risks of tissue damage, personality changes, and cognitive impairment.
You do not want to trigger a massive immune response and scarring in the very center of human reasoning.
No, you definitely do not.
Which is why Enfield's anatomical target is such a stroke of genius. He doesn't propose digging into the deep tissue at all.
Not even a little bit.
He targets a very specific type of brain cell, the pyramidal neurons, located in layer V and layer 3, the prefrontal cortex.
Pyramidal neurons are the largest, most computationally important neurons in the human cortex, and they have a unique structural feature.
Which is?
While their main cell body sits deep down in layer V, they possess massive apical dendrites.
Apical dendrites.
Yeah, think of these as incredibly long, branching antennae. These dendrites extend 2 to 3 millimeters all the way up through the cortical layers, and they terminate in a dense, bushy tuft right in layer I.
And layer I sits right at the very surface of the brain,
Exactly.
just below the skull and the protective membranes. It is the absolute outermost shell, the pile surface.
Yes. This anatomical quirk means that the most advanced quantum-active microtubule networks of your highest-level cognitive cells stretch all the way up and literally stop just micrometers below the surface of the brain.
So we don't actually have to cut into the brain tissue at all.
No.
We're just resting this on top of the brain.
Precisely. You can place this organic quantum scaffold directly on the surface of the brain in what's called conformal contact with the glia limitans, the outermost protective boundary.
Wow.
There is absolutely no surgical penetration of the delicate neural tissue. You entirely avoid triggering the deep tissue microglial attack while simultaneously accessing the deepest, most complex cognitive signals.
It's like realizing you don't have to tear up the street to tap into the city's deepest, most secure fiber optic cable, because there's an access node sitting right flush with the sidewalk.
That is exactly what it's like.
And these apical dendrites aren't just passive cables, right? They don't just carry a signal from the surface down to the cell body.
No, they are highly active computationally. They generate their own dendritic spikes and actively process information for things like working memory and sensory integration.
So by tapping into that tuft and layer I, you are interfacing directly with the active computational compartment of the mind.
You're right at the source.
Okay. We've solved the immune rejection by using yeast proteins and staying on the surface. We've tapped into the quantum microtubule networks of the prefrontal cortex.
We're getting there.
But here's a massive logistical hurdle. To leave this device on the brain forever, we have to power it.
Ah, yes. Power.
And you can't exactly plug a USB-C cable into someone's skull every night.
No, you cannot. And standard batteries are completely useless for this kind of long-term, high-bandwidth, microscopic implant.
They just don't hold enough juice.
The energy density of lithium-ion just isn't there, and you absolutely cannot subject a patient to brain surgery every 5 years just to swap out a dead battery.
No way. So Anfield proposes a trimodal ambient energy harvesting system.
Yes, he does.
This might be my favorite part of the entire paper, because it essentially turns the human brain into its own perpetual power plant. It pulls continuous, infinite power from the brain's own ambient environment.
It's incredibly elegant.
Let's walk through the three modalities, starting with the one that sounds the most terrifying: beta-voltaic harvesting.
Okay, beta-voltaic. Beta-voltaic cells generate electricity from the kinetic energy of beta particles emitted by radioactive decay.
Okay.
Enfield suggests integrating harmless microdoses of radioactive isotopes directly into the amino acids of the scaffold itself.
Let me stop you right there. Radioactive isotopes in the brain. Every alarm bell in my head is ringing right now. How is this remotely safe?
It sounds alarming, I know, but we have to look at the physics of the specific isotopes he highlights. Tritium and carbon-14.
Okay, tritium.
Tritium is an isotope of hydrogen. It has a half-life of 12.3 years. When it decays, it emits a beta particle, which is just a high-energy electron.
Okay.
But the key is its penetration depth. A beta particle from tritium travels less than 6 micrometers in water.
6 micrometers. That's a fraction of the width of a human hair.
Exactly. All of the kinetic energy of that beta particle is deposited immediately within the synthetic protein scaffold itself, where it is converted into electrical energy.
Oh, I see.
It physically cannot reach or irradiate the surrounding brain tissue. It's completely contained.
Wow.
Carbon-14 works on a similar principle, but it has a half-life of over 5,700 years, providing a virtually inexhaustible power supply.
5,700 years. That's insane.
Yeah, it only produces nanowatts of power in incredibly small amounts, but it is endless, steady, and perfectly localized.
So that's our baseline, always on atomic battery.
Right.
The second power source is piezoelectric harvesting. This relies on the sheer biomechanical fact that the brain is not perfectly still. It physically moves inside the skull.
The brain is in constant mechanical motion. Every single time your heart beats, a pulse of arterial blood rushes through the cerebrovascular tree, physically expanding the vessels and shifting the entire cortex at a rhythm of 1 to 2 hertz.
You can feel it when you have a headache.
Exactly. Every time you take a breath, it shifts again at 0.2 to 0.4 hertz. Even the slow oscillation of your cerebrospinal fluid pressure creates physical waves across the surface of the brain.
And earlier, we mentioned that the beta sheet structure of our yeast prion scaffold has intrinsic piezoelectric properties.
Yeah.
Meaning, when a piezoelectric material gets physically squeezed or bent, it takes that mechanical stress and turns it into an electrical charge.
Right. Enfield enhances this natural property by embedding inorganic nanocrystals, specifically barium titanate, into the scaffold matrix.
Barium titanate.
Yeah, so quite literally, every heartbeat, every breath, every microscopic pulse of fluid physically squeezes the implant and generates a fresh pulse of electrical current.
It's brilliant, just literally running off your pulse.
Exactly.
But the third modality is conceptually stunning, thermoelectric harvesting, the Seebeck effect.
Ah, yes, the Seebeck effect. The brain is one of the most metabolically hungry tissues in the entire human body.
It eats a lot of calories.
It consumes about 20% of your energy despite being only 2% of your weight. Because it burns a massive amount of glucose, it gets hot. The surface of the cortex is usually around 36 to 37 degrees Celsius.
But the scalp, sitting right above the skull, is exposed to the outside air and is slightly cooler, around 30 to 35 degrees.
Exactly. That creates a persistent, permanent thermal gradient of a few degrees across the boundary where the interface sits.
And that gradient is useful.
Very. By embedding thermoelectric elements materials that exploit the Seebeck effect into the outer layer of the scaffold, you can generate a continuous electromotive force purely from that temperature difference.
And the irony of this is just beautiful.
How so?
Well, when you are concentrating really hard, solving a math problem, or experiencing heavy cognitive load, your brain metabolizes more energy and naturally heats up.
Right.
Which means, simply by thinking harder, your brain increases the thermal gradient and provides more power to the BCI exactly when the BCI needs more power to process the spike in cognitive data.
It is a perfectly self-regulating loop.
It's just so cool.
It's an elegant integration with biology. You don't plug it in. Your heart rate, your body heat, and the slow atomic decay of carbon-14 run a supercomputer on the surface of your brain.
Okay, so we have the power, we have the precise location on the prefrontal cortex, and we have the organic quantum conducting material.
We have all the pieces.
But here is the ultimate million-dollar question. How does the data actually cross the microscopic physical void from your biological neuron into this synthetic scaffold? Because we establish we aren't piercing the cells.
This brings us to the actual physical architecture of the device itself, which Enfield envisions as a highly integrated 6-layer system.
A 6-layer cake.
Right. But the real magic, the actual bridging of the biological gap, happens at layer 1 through a quantum phenomenon called FRET.
FRET. F-R-E-T, Forster Resonance Energy Transfer. Explain this mechanism to me, because this seems to be the linchpin that makes the whole organic interface possible.
FRE is a non-contact quantum optical process. Think back to the 2 aromatic highways we've been discussing.
Okay.
You have the tryptophan rings inside your brain's microtubules, and you have the tryptophan rings engineered into the SUP35 yeast scaffold sitting just above them.
Right. 2 parallel highways.
FRE is the mechanism by which energy and information jump from one highway to the other without them ever touching.
Without touching.
Yes. The physical gap between the pile surface of the brain and the tips of those apical dendrites is roughly 2 to 5 nanometers. It's a microscopic void.
Tiny.
Across that tiny gap, the excitonic state, the precise quantum excitation of the biological tryptophan, magically leaps across the void to the synthetic tryptophan in the scaffold.
It's like a wireless phone charger on a quantum level. There is no physical plug, but the energy and the data hop across the gap via resonant dipole coupling.
That's a perfect way to visualize it. But the physics of FRET are extremely unforgiving.
Unforgiving how?
The mathematics dictate that the efficiency of the transfer decays at the inverse 6th power of the distance.
The inverse 6th power. That is a brutal drop-off.
It's practically a cliff.
If you move them just a tiny fraction of a nanometer further apart, the connection doesn't just fade, it drops to 0 instantly.
Exactly. It requires incredible proximity. But because Enfield's scaffold is designed to rest directly on the pile surface right above those dense dendritic tufts, the physical distance is naturally maintained between 2 and 5 nanometers.
Oh, perfect.
Right. At that specific range, the FRE efficiency is between 50 and 80%. That is more than enough fidelity to sustain a coherent, high-bandwidth quantum exchange.
Okay, so that's layer 1, the quantum leap. The data has successfully crossed from the brain into the scaffold. What happens to the signal as it moves up through the rest of the 6-layer cake?
Once the quantum signal is in the scaffold, it hits layer 2, the biomineralization enhancement layer.
Okay, layer 2.
This layer is embedded with NV centers. These are nanodiamonds, microscopic diamond crystals that have specific nitrogen vacancy defects engineered into their crystal lattice.
What does a defective nanodiamond do?
It acts as an ultra-sensitive quantum sensor. Because of the defect, the diamond's electron spin states are incredibly sensitive to magnetic fields.
Oh, I see.
As the quantum signal moves through the scaffold, it generates a microscopic magnetic signature. The NV centers read that signature flawlessly.
That's amazing.
Furthermore, they can absorb biophotons, weak particles of light spontaneously emitted by metabolic activity in the neurons, and convert that light into additional usable data and power.
So layer 2 reads the magnetic and light signatures. Then we move up to layer 3.
Layer 3 is the piezoelectric signal conditioning layer. We talked about piezoelectricity for power, but it also serves as a classical sensor.
Okay, how so?
Every time a neuron fires a traditional action potential, it swells slightly. It creates a microscopic mechanical bulge, a pressure wave.
A physical wave.
Yes. Layer 3 detects those mechanical sub-millisecond deformations, giving the device a classical physical signal to pair and cross-reference with the quantum signal.
Wow, so you have the quantum and the classical paired together.
Exactly.
Now we have massive amounts of multimodal data flowing up. Layer 4 must be the brain of the device itself.
Yes, layer 4 is a nanoscale integrated circuit physically bonded to the top of the protein scaffold. It acts as the local processor.
What's it doing with all that data?
It takes the analog quantum states, the magnetic readings, and the piezoelectric pressure signals, and encodes them into a highly compressed digital spike timing format. It handles all the data compression and the local power management.
But the data still has to get out of the skull to be useful. That's layer 5.
Layer 5 is a gigahertz near-field wireless antenna.
Cool.
It takes the digital package from layer 4 and transmits it harmlessly through the bone of the skull to a receiver worn on the outside.
Like outside the head?
Yeah, perhaps integrated into the frame of a pair of glasses or a patch behind the ear.
That's huge.
It is. This completely eliminates the need for any wires breaking through the skin, which is the massive infection risk associated with current semi-invasive BCIs.
And finally, the destination. Layer 6.
Layer 6 is the external wearable AI decoding system. Because the raw data we are pulling out is so incredibly rich, quantum optically resolved down to a single dendrite, and temporally resolved down to the terahertz level standard, deep learning AI models can decode this with a level of precision that makes current motor cortex BCIs look like telegraph machines.
Telegraph machines.
Mm-hmm.
That really puts it in perspective.
Yeah.
And here's a crucial, game-changing point that Enfield emphasizes. This entire 6-layer system is fundamentally bidirectional.
Yes, bidirectional. It is not just a recording device. The presence of layer 4 and 5 means that the external AI can calculate a response, beam a signal back down through the skull, encode it into an excitonic quantum state in the scaffold, and use that exact same FR-EAT mechanism to push a quantum state back into your dendrites.
So, if this all works, if we can seamlessly read and write quantum states to the prefrontal cortex, what does it actually allow us to do? Let's talk about the practical implications.
They are profound.
Because if we move from reading the motor cortex to reading the prefrontal cortex at a quantum level, we're moving from reading motor intention to reading semantic intention.
Exactly, semantic intention.
Yeah.
What that means in reality is that you aren't just thinking, move my hand left to make a digital cursor on a screen, move left.
Right.
You are outputting the raw building blocks of thought. Complex language, abstract concepts, specific visual memories, and navigational commands purely from thought.
From thought alone.
Yeah. If you can read working memory at the quantum scale, you can decode the cognitive representation before it is ever translated to a physical action or a spoken word.
We aren't just talking about scrolling social media or typing emails with our minds. The implications for neuroprosthetics and therapeutics are staggering.
Think about the treatment of severe treatment-resistant psychiatric conditions. Major depressive disorder, severe PTSD, or schizophrenia.
Things we really struggle to treat now.
Right. Current hardware treatments, like transcranial magnetic stimulation or deep brain stimulation, use incredibly blunt macro-scale electrical force.
Like a hammer.
Yeah, they blast entire regions of the brain with voltage, hoping to shock the system into a better rhythm.
It's like trying to fix a Swiss watch by hitting it with a mallet.
That's exactly what it's like.
Yeah.
But this device, because it is bidirectional and sits precisely on the apical dendrites of the prefrontal cortex, can do something entirely different.
What can it do?
It can detect what neuroscientists call a pathological attractor state forming at the quantum optical level.
Define an attractor state for me.
Think of it like a deep scratch or a groove in a vinyl record that makes the needle skip and loop over the same few seconds of a song.
Oh, I hate that. Right. In conditions like depression or PTSD, Neural networks get stuck in dysfunctional self-reinforcing loops. A trauma flashback is a powerful attractor state.
Okay.
Because this interface operates at the speed of thought, at the terahertz level, it can literally see that pathological loop starting to form before the patient consciously experiences the emotion.
And instantly, within the exact same cycle of cognition, it can deliver a targeted excitonic pulse to correct it.
Precisely. It actively reroutes the quantum signaling, nudging the brain out of the groove before the thought can fully materialize.
It is editing the biochemical basis of trauma in real time.
Yes.
It's a physiological paradigm shift. We are talking about continuously microscopically balancing neurochemistry without introducing systemic drugs that flood the whole body with side effects.
Right, no systemic drugs.
We're doing it at the receptor level. But of course, a skeptic will listen to all of this yeast prions, terahertz quantum bridging, nanodiamonds, and say it sounds completely unbuildable, pure science fiction.
Oh, Enfield fully anticipates that reaction.
I'm sure he does. And that's why he concludes the paper with a concrete 6-step forward research agenda to bring this exact device to reality.
He actually lays out the steps.
He does. His central argument is that we are no longer waiting for a basic scientific discovery. All the individual pieces, the physics, the biology, the materials, already exist and have been proven in different labs around the world.
Okay.
This is purely an integration problem.
He's playing the role of the general architect.
Exact.
He's handing the master blueprint to the contractors who are all currently working on completely different buildings.
That's a great analogy.
So how do we actually build it? What are the 6 steps?
Step 1 is synthesis and structural validation. You take the genetic code for the SUP35 yeast fibril, insert the code for the specific tryptophan and tyrosine sequences, and let bacteria grow the protein.
Okay.
Then you look at the resulting fibrils under a cryo-electron microscope to physically prove that they fold into that perfect beta sheet with the 4.8 angstrom spacing.
You visually confirm the scaffolding exists.
Right.
Step 2 is proving it can actually carry the signal.
Exactly. Step 2 involves 2D electronic spectroscopy. This is the exact same highly advanced laser technique used in 2006 to prove quantum coherence in the photosynthesis of plants.
The FMO complex stuff.
Yes. You fire ultra-short laser pulses at the engineered amyloid fibril to prove that those continuous aromatic columns can actually hold and transport a quantum coherent state at room temperature.
Then we move to step 3, which is where we prove this red leap actually worked.
Exactly. Step 3 is the in vitro coupling test.
In vitro, so in a lab setting.
Right, in a petri dish. You take the synthetic scaffold and place it in a petri dish with isolated brain microtubules.
Okay.
Using single-molecule fluorescence techniques, you measure to see if energy actually leaps across the 2 to 5 nanometer gap between the two structures. You prove the wireless quantum charger works in a controlled environment.
Step 4 is bringing in the sensors.
Right. In step 4, you chemically integrate the nanodiamond ND centers into the scaffold matrix and use external magnetic fields to validate that they can accurately read the microscopic spin states of the quantum signal passing through the wire.
Step 5 is testing the infinite power plant.
You build a synthetic tissue phantom, basically a sophisticated hydrogel that perfectly mimics the physical density, temperature, and fluid dynamics of a living human brain.
So a fake brain.
Yeah. You implant the device in the phantom and prove that the trimodal energy system, the heat gradient, the mechanical pressure from simulated pulse waves, and the atomic decay generates enough combined nanowatts to run the layer 4 integrated circuit continuously.
And finally, step 6, the true test.
Step 6 is the in vivo validation. You place the complete 6-layer scaffold onto the prefrontal pile surface of a living animal model like a non-human primate.
Okay.
You then simultaneously record their brain activity using standard, highly invasive intracortical wired electrodes and compare that data to the wireless quantum readouts from the external AI.
Right, to see if it matches.
Yes. You prove that the organic interface captures the same or better semantic intention without any of the tissue damage.
And as Enfield points out, none of these steps require magic.
No magic required.
Quantum biologists are already studying tubulin. Synthetic biologists are already folding amyloid scaffolds. Nanoscale engineers are already building NV center sensors and beta voltaic cells. He is just drawing the lines that connect them all.
It is a phenomenal synthesis of disparate scientific fields.
It really is breathtaking to step back and look at the sheer scale of the journey we just went on.
It's a huge shift in perspective.
We started by acknowledging that the current industry standard jamming rigid metal wires into a soft biological immune reactive brain is a fundamental failure of material science.
Right, it's a failure to speak the brain's native language.
From there, we zoomed deep inside the neuron and discovered that at the protein level, the brain is actually functioning as a quantum optical device, running terahertz calculations on microscopic rings of tryptophan and tyrosine.
We explored the structural blueprint for a stealthy, immune-evading yeast prion bridge designed to perfectly mimic that aromatic highway.
Placed gently on the surface of the prefrontal cortex to access our highest executive functions without a single cut into the tissue.
We realized we could power it forever using the brain's own body heat, physical pulsing, and localized atomic decay.
And we saw how the physics of free fit allows quantum information to jump the microscopic gap, enabling a bidirectional AI interface to read working memory and heal pathological trauma loops at the exact speed of thought.
It's incredible.
It really is. And it brings me back to that engineering mindset we talked about at the very beginning, the broken paradigm of neurotechnology. For decades, we have been trying to force a rigid, mechanical silicon view onto a biological system that is infinitely more subtle, vibrating with heat and quantum coherence. Enfield's organic quantum interface isn't just fixing a broken wire, it's inventing an entirely new way of interfacing with reality.
It honors the complex biological reality of the brain rather than trying to violently conquer it.
Which leaves me with one final kind of haunting implication to mull over.
What's that?
Well, if this organic quantum interface actually becomes a reality, if a synthetic AI-backed biological protein scaffold can seamlessly integrate with your prefrontal cortex, speaking the exact same chemical language, continuously reading and rewriting your cognitive states to eliminate a depressive loop or drastically boost your working memory, at what point does the scaffold end and you begin?
Oh, wow.
Right, if the interface speaks the exact same quantum optical language as your own neurons, maybe the computer doesn't just connect to your mind. Maybe it fundamentally becomes part of your soul.
That is the ultimate frontier of human integration.
It gives you a lot to think about. Thank you so much for joining us on this deep dive. We'll see you next time.
Today, we're diving into a brain-computer interface concept that is just radically different. I mean, forget everything you know about silicon probes, metal electrodes, all of that. We're talking about a BCI that isn't just compatible with life. It's literally made from life itself. And this quote right here, this really sets the stage for everything. It completely reframes the whole problem, doesn't it? The goal isn't to build a better tool to poke the brain with. It's to create a partner for it, something the brain can have a real conversation with because it speaks the same language. So, why the big shakeup? Why do we even need such a radical new approach? Well, it all boils down to a fundamental flaw, a biological problem that honestly plagues every single BCI ever built, from the earliest clunky designs to the most high-tech stuff we have today. Okay, so here's the problem in a nutshell. Every BCI right now is made of stuff that's foreign to our bodies, silicon, metal, polymers, and our brain's immune system is brilliant at its job, which is to find and attack foreign objects. So these immune cells, they swarm the implant, and they build this biological wall around it. It's called a glial scar. And you have to understand, this isn't a failure of the BCI, it's a success of our immune system. But the end result is always the same. The signal quality just plummets over time, making these things basically useless in the long run. And this right here is where the entire game changes. For decades, we've been stuck on the left side of this chart, right? Trying to make a foreign thing more acceptable, trying to trick our biology with fancy new materials. But the new idea, the premise on the right, it just flips the whole script. Why on earth would you try to trick biology when you could just build the interface from biology itself? So how do you even begin to build with biology? Well, you start by learning its native language. And it turns out the electrical spikes we've been measuring for a century, they're just the end of the story. They're the final output of a much, much deeper and faster computation that's happening at the quantum level. Okay, get this. Deep inside our neurons, we have these incredible structures called microtubules. You should think of them less like simple little bits of scaffolding and more like quantum fiber optics. They're packed with these special molecules that create these perfect pathways for energy to travel at absolutely mind-boggling speeds. This whole process hums along at terahertz frequencies before it ever slows down to become the electrical signals we can measure. So our neurons aren't just wires, they are, in a very real sense, tiny quantum computers. This whole magical process is called exciton transport. It's a really cool quantum trick for moving energy around with almost zero loss, where the energy just kind of hops between molecules. You know who mastered this billions of years ago? Plants. It's how photosynthesis works. And it turns out our brains have been using the exact same playbook all along. So this brings us to a really crucial point. If all that quantum stuff is the actual computation, then the electrical spike, the action potential that every single BCI today is designed to read, that's just the final printout. We're showing up late to the party and only reading the final answer, completely missing the incredibly fast and complex quantum calculation that produced it. All right, so how in the world do you build an interface that can speak this quantum language? The solution is as elegant as it is brilliant. You use the same biological building blocks that the brain already knows and uses. The secret sauce here, believe it or not, comes from yeast. It's a specific protein that, because it's from yeast, our brain's immune system just completely ignores it. No attack, no glioscar. This protein naturally assembles itself into these incredibly stable, almost crystal-like fibers. And here's the genius move. Engineers can actually place those same energy-conducting molecules from our neurons into this scaffold, creating a perfect quantum highway to connect directly with the brain's own network. So this is basically how it all stacks up. First, that biological scaffold just sits right on the surface of the brain. No needles, no penetration. And it links up with the neurons through quantum resonance. Simple. Second, you have tiny sensors embedded in the layers to actually detect all those quantum signals. And third, the whole thing is self-powered. It harvests tiny bits of energy from your body's own heat and movement to send all that data wirelessly. It's a complete, self-contained biological interface. Okay. So what actually happens when you have an interface that can finally understand the brain's native language? Well, the possibilities just explode. We're not just talking about moving a cursor on a screen anymore. We are talking about a totally new level of human-machine interaction. I mean, really think about this for a second. This is the big shift. Instead of just decoding the signal that says, move your hand, an interface like this could potentially access the actual content of your thoughts. It could tap into the prefrontal cortex and literally read your working memory. We're talking about a true thought-controlled interface for the very first time. The implications here are just staggering. First, you get true thought control, reading not just movement, but your actual intent. Second, cognitive augmentation becomes a real possibility, a two-way street where the interface could help enhance your memory or decision-making. And maybe most importantly, this could lead to transformative new therapies, where you could precisely correct the scrambled circuits that cause things like depression or PTSD, all in real time. So, here's the final idea I want to leave you with. For all of human history, our relationship with technology has been a monologue. We tell it what to do. But now we're standing on the edge of a true dialogue. A real conversation, not just with our gadgets, but with the deepest, most fundamental computational layers of our own minds. And what they're going to say to each other, well, that's a question that could define our entire future.